Hello everyone and welcome to Pocket Size Bio with Dr. Casco. In today's video we're going to look at chapter 6 of the Campbell Biology textbook. This is a tour of the cell. Now this is a very large chapter. It has 8 sections. We're going to break down this video into 2 parts, sections 6.1 to 6.4, and then the second part 6.5 through 6.8. So let's get to it. You can see the key concepts that we're going to take a look at. So we're going to start off talking about microscopy and a little bit of biochemistry to study cells. And then we're going to get into eukaryotic cells and prokaryotic cells and some general features. And then we enter the genetic instructions phase and the nucleus, getting into the endomembrane system for this part of the video. Taking a look at this kind of intro picture here, figure 6.1. The cell is an organism's basic unit of structure and function. Many forms of life exist as single-celled organisms, such as the paramecium shown here. Okay, so this is a single-celled organism. Larger, more complex organisms, including plants and animals, are multicellular. In this chapter, we focus mainly on eukaryotic cells with a nucleus. Okay, so that's one of the defining features of eukaryotic cells. As we take a look here, there's a very nice diagram here of a plant cell, right? How does the internal organization of eukaryotic cells allow them to perform the functions of life? So internal membranes, they divide a cell, as we see here, this plant cell, into compartments, right? These compartments enable specific chemical reactions to occur in isolation. So there's no interference between other chemical reactions that are occurring. So that improves the overall efficiency of the cell. So there's several locations, specific areas, specific structures within the cell that undergo specific processes. Generally, you break down this stuff into energy and matter transformations, genetic information storage and transmission, and interactions with the environment. So we're going to take a look at all these organelles, all these structures, and the processes that occur within these structures overall to make the cell one functional unit. Let me go to the next section here from the beginning. So if you see here, I have basically a duplicate of my pages here, so you can take a look at the figures as I go along with the notes. I'll be moving these figures along so it can be easier for you to be looking and kind of being in sync with the notes here. We start off with section, of course, 6.1. Biologists use microscopes and biochemistry to study cells. As we all know already, microscopes, they're instruments, right? Scientific instruments that extend the human visual perception. Invented in the 1590s. Okay, we constantly in high school, in our schooling, we always contribute this idea of microscopy with Robert Hooke, because he coined the term cells. As he was observing cell walls on bark, right, on oak bark more particularly. He was basically the one that gave these little cells the term cells. Okay, we have Antony van Leeuwenhoek. He also observed living cells. It did many experiments where he basically observed some bacteria, little protists, red blood cells within human tissues, right? So microscopy and the advent of microscopes gave rise to this whole world that previously nobody knew about. So it was incredible to have this development, right, of machinery, of technology. And ultimately, we have this light microscope, right, where visual light passes through a specimen, right, through glass lenses. And the lenses bend light ultimately in such a way that the image is magnified. So you see here a little just general generic picture of a microscope kind of breakdown. Within the inside of the microscope, you have a light source and you have several lenses, right? And you have your specimen on the stage, right, on the microscope slide that you've prepared. And this light source will beam light through the specimen, and it will refract and bend in such a way that you will be able to see the image much larger than what you would normally see with your naked eye. The idea being microscopes magnify an image, right, a specimen that you're visually seeing. In terms of microscopy, there's 3 parameters the biologist always needs to take a look at: magnification, resolution, and contrast. And as we all are familiar with, magnification is probably the most common parameter when you think about microscopes. It's essentially increasing the size that you're observing the organism or that specimen, right? It's the ratio of the object size relative to the real size. Light microscopes can magnify about 1000x, some of the stronger ones, right? There is a kind of trade-off that as the magnification increases, the object details cannot be seen as clearly, right? So yes, you want to enlarge the image, but you're going to tend to see it more blurry. So that's a kind of sacrifice you're having with magnification, which leads to this next parameter, resolution. Now, resolution is a measure of the clarity of the image, right? It's the minimum distance between 2 points, which can be separated and distinguished apart. Right? So it's that clear refinedness that you can see the structures of a specimen or whatever sample you're looking at. And contrast. So contrast deals with a play within the brightness of the amount of light you're using in the dark areas of an image of a specimen, right? And there's different techniques that you can do and perform on the specimen to enhance how it visually looks, okay? So you have methods for enhancing, including staining and labeling structures that we're going to see a little bit later, okay? One thing that is quite the limitation of light microscopes is that organelles, you cannot be observed clearly. Membrane enclosed organelles, right, structures, cannot be observed by light microscopy due to resolution limitations. We can see the nucleus if we stain it, right, but we won't be able to see the phospholipid bilayer just because they're too small to pick up by the light microscope. Okay, so because of this, my biologists and engineers needed to find a way to be able to observe these structures, right? So you get the electron microscope in the 1950s. And what happens in the electron microscope is that you use, instead of light, you're using a beam of electrons. They pass through the specimen, or they're reflected the specimen from the surface onto a detector. And that detector will process whatever electrons it's capturing onto an image. And that image is what we would see on a computer and whatnot. In terms of electron microscopes, there's generally 2 types. So you have a scanning electron microscope and you have a transmission electron microscope. The scanning, it's used primarily for topography, seeing the surface of the specimen. This type of microscope generally coats the specimen with a film of gold, so you can have a more efficient reflection of electrons. The beam is excited, so the beam of electrons, right, excites electrons on the surface, and then those secondary electrons that are being excited get detected. You have transmission electron microscopes, and transmission, I like to tell my students, right, transmission T is meaning that you can see through the specimen. Scanning, surface, so starting with an S, just a little way to help you remember, right? Scanning, surface, transmission, seeing through the specimen. So if you see through, then you can see the internal structures of a specimen, even a single cell. Very, very thin sections you can create and see all the organelle and all the structures of the cell or whatever it is you're you're examining. These cells, again, or these specimens, are also stained with heavy metals to enhance electron density, and that can help pick up, again, from the detector and have a high resolution of whatever it is you're observing. In general, electromagnets help you kind of resolve and magnify more rather than just light. You don't use light in these cases. So electromagnets are used to bend the beam that you're shooting out, and then that can help cause a general focus, resolving, right, the image you're seeing. Again, like I mentioned before, electron microscopes have been used to make subcellular structures visible that were impossible to see with the light microscope. That doesn't necessarily mean that the light microscope is not useful. It still has its advantages, right? You can study living cells with the light microscope as opposed to the electron. Sorry, I probably confused that. So the light microscope still has advantages, right? You can study living cells as opposed to the electron microscope, where you need to essentially preserve and kill the organism, right? It can't be alive because essentially it's in a vacuum, and you don't have that much noise. Okay, light microscopes have gotten a major upgrade within these past decades, if I can say. Technically, where you can use fluorescent staining, and fluorescent staining, along with laser microscopy, can enable you to see incredible detail of the specimens you're observing, right? Such as confocal deconvolution microscopy can even produce 3D images, right? Super resolution microscopy has allowed scientists to break the resolution barrier just because we're innovating with lasers that we're shooting onto the specimen to get a more clear, ultra resolution samples, right? Images. There's a new type of transmission electron microscope called cryo-electron microscopy. It allows specimens to be preserved at extremely low temperatures. So this is a great thing because generally, in order to process specimens or slices that you've done, sections that you've done of a particular sample, you need to preserve it. You need to use a fixative, and the fixative can destroy or alter the properties of proteins, of certain structures, of membranes, and that can give you a lot of noise. That can reduce the efficiency of fluorescent staining. So by avoiding using preservatives, you can detect more effectively the natural cell state, protein localization, and the complex and the overall structure of the sample that you're seeing better, right? So microscopes, they're the most important tools in cytology, cell biology, right? They study cell structure, and by that understanding, right, you can understand the function, and you can see the structure better. But in order to actually understand the function of how certain tissue works or how a cell works and whatnot, you also need to integrate biochemistry, right, which is the study of chemical processes of cells. So you need to get inside and get some quantification of proteins, of mRNA, and see what activity is going on in the cell, right? Observing just a preserved sample isn't enough. You need to figure out what exactly it's doing at a particular moment. The panel that you see here is just relative ranges of structure sizes and how, our limitations with our eyes, we can see a certain size, right? The unaided eye can see, pretty small. You're good 120 approximately, 120 micrometers, in size, maybe a human egg, a frog eggs, or 1 millimeter, chicken, and everything larger with that. But, as we go smaller than that, it's incredibly difficult for you to see something like this, like a cell, like a bacteria, like a virus, definitely not with the unaided eye. So of course we need light microscopes, and then furthermore electron microscopes that their size is just so incredibly small, going down from the micrometers down to the nanometers and in point nanometers, right? So the advent of microscopes has been a breakthrough in biology. Let me continue on. Let me also move this to this example here. I'm going to move this to the same, okay? Figure 6.3 is a very nice depiction of the different types of microscopy, right? Now, I won't go into so much detail with this, but you can definitely look at these on your own and see how the different images can provide different information, right? You have bright field microscopy. This is light microscopy. Now, light microscopy doesn't necessarily mean that it only uses light. It could have help with, again, lasers, fluorescence, and so forth, right? So you have bright field microscopy. We have images like this. Let me just zoom in a little bit. Phase contrast. So this is what I was referring to as the contrast. You could see the dark areas and get kind of a pseudo 3D structure within that light simple microscopy. Interference contrast, or differential interference contrast microscopy. Again, you can see something similar in terms of that 3D structure, especially those organelles and those structures that are quite large, okay, due to density differences. For fluorescence microscopy, you could see incredible detail of individual structures just because you're using immunostaining, right? You're using antibodies which bind to very specific structures that you want to see. And there's different types of antibodies that could label different things. Here you can see the cytoskeleton being labeled, the mitochondria being labeled, the nucleus, and so forth. So there's so many different types of antibodies. Depending what you would like to see, you would purchase and order one from a biological company. Confocal, deconvolution, and super resolution are again, this is a level up in microscopy where you would use lasers to essentially scan at the micro level different sections of even of a cell, right? Or a tissue sample that you're seeing, that you can actually get an actual 3D structure on the computer. And you can see it and you can rotate it and you can see what's going on within the whole sample, okay? So it's a better way to visualize something that you're trying to annotate and see where it's being found. For example, a particular protein, a growth factor, for example, within a tissue sample, you can see where it's actually localized in the tissue that you're seeing. Okay, and then electron microscopy. Here's an example of the scanning electron microscopy that you can see in incredible, beautiful detail. Again, this is still staining cilia within this particular sample of the trachea. That same tissue sample, if you're seeing it under the transmission electron microscope, you can see how each individual cilia is kind of composed of. Okay, and again, this is what I was mentioning as to cryo-electron microscopy, where you can even create 3D images that is data processed through all the detections of electrons being beamed back to the software and to the computer, and so forth. Let's continue on. Let's talk about cell fractionation. This is a technique used in separating cell structures and studying the functions that these structures can have. And what happens here, as you see in the panel here, in the diagram, this is done by taking cells apart and separating the components through centrifugation, right, or through a centrifuge. So generally, cells are blended together. Let's say you have some particular sample of tissues that you want to harvest. You homogenize the sample into this homogenate, and you begin centrifuging at different speeds. Okay, and as you may know, right, when you centrifuge something, you have a pellet, that's solid material that is at the bottom. And then you have a liquid phase, which is referred to as a supernatant, right? And what scientists do is that supernatant, they transfer to a new tube, and it's poured into a new tube, and then you would centrifuge again at a different speed, right? In this case, let's say you have 1000 G of centrifugation, and then the second, tube would be at 20,000. Then you would repeat the process. You'll transfer the supernatant to 80,000 G and to see what you would collect on that little pellet there. And at different Gs, you would collect different material, right? So at the slowest one, you get a pellet that's rich in nuclei and cellular debris. As you start to increase the centrifugation force, right, you have a lot of mitochondria and chloroplast. In this case, if you have a plant, right, at 20,000 Gs, and then you get microsomes, right, so pieces of plasma membrane, internal membranes, and so forth. One of the highest ones being 150,000 Gs, you get a pellet rich in ribosomes. So depending what you want to observe, that's what you would do to collect that particular structures and specimens, right, and samples that you want to analyze and process. So in summary, lower speeds yield larger components, higher speeds yield smaller components. Moving to section 6.2, eukaryotic cells have internal membranes that compartmentalize their functions. So again, we know that the cells are the basic structural and functional unit of every organism. There's two distinct types. We have prokaryotic cells, we have eukaryotic cells. Prokaryotic cells can be broken down into subgroups, bacteria and archaea, right? Eukaryotic cells, you have several kingdoms that you have this type of eukaryotic cell. You have the kingdom Protista, fungi, animalia, plantae, and of course they have their differences between prokaryotic and eukaryotic cells. The image you see here is an example of a prokaryotic cell, okay, and some structures that you might be familiar with, right? They do not have a nucleus. This is a rod-shaped bacteria, a bacilli, and you have a bacterial chromosome that is in this nucleoid region. You have ribosomes spread out throughout the structure. Some bacteria have little fimbriae, right, that enables them to attach to surfaces, okay? Essentially, fundamentally, you have plasma membrane. Every single cell needs to have a plasma membrane, okay? Cell wall for particular cell types, rigid structures outside of the plasma membrane, and furthermore, bacteria can even have a glycocalyx, which is an outer coating of many prokaryotic cells, which is a capsule or a slime layer. Some bacteria can move around with a flagella, and if you see the bacterial cell under an electron microscope, you would observe something like this, right? 0.5 micrometers in distance. This is an incredibly small organism. It is important to note that you should memorize all of these structures. One of the defining differences between eukaryotic and prokaryotic, as I mentioned before, is the presence of a nucleus. Eukaryotic means true nucleus. Prokaryotic means before the nucleus formed. Types the membrane organelles. So then you have the region between the nucleus and the plasma membrane. It is called the cytoplasm. Within the cytoplasm, it's further broken down into subparts. You have the cytosol, which is the actual fluid part of the membrane of the cytoplasm, and then you have the organelles that are suspended within the cytosol. Prokaryotes, very important. Okay, always remember they lack organelles, right? So they don't have, moreover, membrane-bound organelles. They do have regions, and this is quite impressive. It's quite interesting that they do have regions where specific proteins are localized. So you can have areas of biochemical pathways, reactants and products interacting together, right? Being made in particular areas within the cell, even though there's no compartments, right? Eukaryotic cells, being much larger than prokaryotic cells, they are able to have compartments which can enable them to be more sophisticated. They can be a lot larger. So because they're a lot larger, they can do several functions. Since they're a lot larger, they can also have differences in structure. Differences in structure will enable them to do a particular function, as we're going to take a look at later on. Smallest bacteria are mycoplasms, 0.1 to 1 micrometer, right? But the typical bacteria ranges around 1 to 5 micrometers. Eukaryotic cells go from 10 to 100 micrometers. Metabolic requirements also impose upper limit to size. This is quite interesting, okay? Metabolic requirements also impose an upper limit to how big you can be. The plasma membrane, okay? So if you take a look here, this is a nice diagram of the plasma membrane, or a section of the plasma membrane. If we were to see an electron microscope image, it would look something like this, right? Of course, this is stained, having a, if you see the scale 0.04, it is incredibly thin, okay? So the plasma membrane is a selective barrier, right? It's a boundary between the inside of the cell and the outside environment, and it's very selective, right? It allows the passage of many materials, of a lot of materials, and some examples are oxygen, nutrients, right? Waste material needs to be leaving the cell, nutrients need to be coming into the cell. And it's very selective in how it does that. For every micrometer square of membrane, only a limited amount of substance can actually pass through per second. So because of this limitation, cells tend to be generally small, right? There's a ratio of surface area to volume that a cell needs to have. Because as the cell size increases, the surface area grows proportionately less. Small cells tend to have a higher surface area to volume ratio. The better the ratio, the more traffic of material you're going to have. Okay? This is the reason why cells are limited in their size. Modifications, you could have changes to kind of bypass this. Modifications in shape can enable the size to increase. So this is a little kind of schematic of what I'm talking about, geometric relationships between surface area and volume. Let's say you have these three situations, right, where you have one small little cell. It doesn't matter what unit it is. Let's say centimeters, right? You have this individual small little cube, right? Total surface area, for simplicity's sake, that a cube has 6 sides, right? So for the total surface area, you basically calculate the height and width of each side. Let's say you have 1 centimeter times 6, right? So height times width times the 6 sides and the number of cells. In this case, we only have one cell. So you have 6 centimeters squared. This larger cube, let's say a larger cell, that is 5 centimeters, will be 150 centimeters squared. Remember, right now we're talking about total surface area. Imagine if you had now cells that were tiny, 1 centimeter, but you had enough of them to be equivalent to the same size, the same volume as this one large cell. Look how big their surface area is, their total surface area is 750. If we calculate the total volume, this is 1 cubic centimeter, this is 125 cubic centimeters, and the volume of this complex of cells is still the same volume. But when you calculate the surface area to volume ratio, which is the surface area divided by volume, take a look at the ratio, the magnitude of the ratio. This small little square has 6. This large cube that is 5 centimeters in length on all sides is 1.2. And this equivalent kind of complex of tissue, right, of cells creating a tissue, is also 6, right? So as you get bigger, your volume ratio, your surface area to volume ratio will decrease greatly. So because of this, cells cannot be large. Continue on. I posted here if you're interested in doing this science skills exercise. For these videos, I just give the theory, right? But in the future, I may come in and provide videos for these exercises as well as multiple choice and critical thinking questions. Another important thing to note is, other than surface area to volume ratio, is the idea of compartments. So now we're essentially talking about eukaryotic cells. Eukaryotic cells, they have an extension, elaborate, arranged internal membranes, right? So it divides the cell into compartments. By having compartments, you can provide different local environments. So by having different environments, specific processes can occur within these little environments, and they could be isolated from other processes, which they could probably come into conflict or are incompatible. Okay, so by having them isolated, you can have two things or more, of course, multiple things happening at once to have a particular outcome. Okay, so membranes also participate directly with reactions. Proteins are embedded within the membranes. Sorry about that. Each membrane has a unique composition, different types of proteins, different ratios of the proteins that you need to be using. Okay, mitochondrial proteins are embedded within the mitochondria to undergo cell respiration. Okay? You're not going to have these proteins in another organelle like a lysosome, right? Because the function occurs in mitochondria. For example, the electron transport chain that we're going to take a look at later. What I'm going to visually show you is just a representation of the animal cell and the plant cell. Okay, all of the structures you see here, I strongly recommend that you remember them, the structure and the function of each individual thing. We're going to go through these one by one, but I just want to show you an artistic depiction of what a typical cell would look like, right? The largest structures being the nucleus, right, surrounded by the endoplasmic reticulum, where you have smooth and rough, and then you can have Golgi apparatus floating embedded along within the cytoplasm, along with mitochondria. If you have a plant cell, you can have chloroplast, and so forth. Everything is sustained together, maintained together by the cytoskeleton, and so forth. There's some additional structures that I'm going to talk about later, but I strongly recommend using this diagram to study all of the different organelles. This is a nice depiction, micrographs, right, of a animal cell, of yeast cells. Okay, this is a surface electron microscope. This is an transmission electron microscope. Okay, take a look. Let me go to the plant cell. Again, same thing. You can pause the video here and take a screenshot if you'd like to help you study. Same situation here. You have plant cells and you have unicellular algae, and this is a transmission electron microscope image of what is occurring within this single-celled green algae here. Chlamydomonas, several chloroplasts that you can take a look at. Let's continue on to the next section here. I'm going to stop the video. So we're going to continue on to 6.3. The eukaryotic cell's genetic instruction are housed in the nucleus and carried out by the ribosomes. The nucleus. We all know, we've been seeing this from middle school to high school until now, even undergrad and so forth. It is an information central, right? It contains most genes, some in the mitochondria, the nucleus. The nucleus is information central, right? It contains most of the genes of the organism. There are some DNA that can be found in the mitochondria or chloroplasts, or both, but in essence, it holds the DNA of the organism. Usually, it is the largest organelle. It's about 5 micrometers in diameter, and it is enclosed in a nuclear envelope. So it's separated from the cytoplasm, and that nuclear envelope is a double membrane. What does that mean? So if we take a look here, you can see, I apologize for the not Casco quality HD image here, but if you were to zoom in to the double membrane, you would see that you would have 2 phospholipid bilayers side by side here. That's what double membrane means. let's continue on. So throughout the nucleus and that membrane, you have perforations. You have little pores that are assembled via a pore complex, as you see here. You can have several structures and proteins that create this little hole, and this pore complex regulates the entry of RNA and protein coming into and out of the cell. The inner envelope, the inner side of the nucleus, is, I want to say, framed or lined with what is called nuclear lamina. And this nuclear lamina is just a network of little filaments of proteins that you can see here. It kind of looks like microscopic gauze that doctors use, gauze. And it helps maintain the shape of the nucleus. Within the nucleus, also you have the nuclear matrix, right? So they're basically protein fibers that are scattered throughout the interior that help also to maintain the shape and also regulate, molecules and complexes, move from one place to another. Talking about the DNA is organized into discrete units called chromosomes, and these structures are what carry that genetic information of the organism, right? We're going to see how that happens in an entirely different chapter. Right now, we're just talking about the structures within the cell. So the DNA strands, think of them as little filaments, little nylon strands, right? They're sometimes coiled with proteins that are called histones. And these histones enable the DNA to wrap around it and fit essentially within the nucleus. We have so much DNA in our cells. They wouldn't be able to fit everything in there if not the help of these proteins. So when you have this kind of loose string form, right, where DNA is wrapped around these little protein complexes, these histone complexes, it is called chromatin. You have this form of DNA when, in general, the cell is not dividing, when it's undergoing DNA expression and so forth. But when the cell is going to divide, it needs to become very compact, right, in a very neat package to enable the whole process to be successful and without any errors. DNA condenses through a lot of coiling, looping, and supercoiling. Okay. Again, this is another thing that we are going to see during mitosis and meiosis. But for now, understand, depending on what stage the cell is in, the DNA will be in a loose form or in a very compact coiled form. Different species have different numbers of chromosomes. Humans have 46, fruit flies can have 8, right? It depends on the species. Within the nucleus, you have a particular area, right, which is referred to as the nucleolus. And this is a mass of densely contained granules and fibers. Okay, RNA is being made here. It is developed into the subunits of the ribosomes, where they, after they're formed, they go and exit the nucleus. Okay, different species have different number of nuclei, and cells won't have the nucleus. Some cells can have the nucleus only at a certain stage of their life, like red blood cells. The nucleus dissolves after it's matured, since it's used for transportation of oxygen. We move on to the ribosomes. Ribosomes are the protein factories. Their complex is made up of RNA and protein. Some of the functions that ribosomes can do is to, primarily ribosomes are used to carry out protein synthesis. It is not membrane bounded. Cells with high rates of protein synthesis will generally have a particularly large number of ribosomes. For example, the human pancreatic cell makes many digestive enzymes, so they have millions of ribosomes. There's two types of ribosomes depending on their location. You can have free ribosomes. They're the ones that are suspended within the cytoplasm. And the proteins made here generally function to be spread out throughout the cytosol and the metabolic processes that are needed within the cytosol. And then you have bound ribosomes. These are the ones that are attached to the endoplasmic reticulum or the nuclear envelope. Proteins made for membrane or for exportation of the cell. Okay, let's take a look at how the ribosome looks like under the an electron micrograph. If you were to take a section of the ER, you would see ribosomes that are free floating here in the cytosol, and then within the endoplasmic reticulum, these little lines that you see here, they are also embedded. Okay, and a depiction of a ribosome actually looks like this. It's composed of two units, a large subunit and a small subunit. A computer-generated model will look something like this, where you would have these RNA complexes and these protein complexes here together. Okay, system graphic of structures. Right. Moving to section 6.4, the endomembrane system regulates protein traffic and performs metabolic activities. So the endomembrane system includes the following group of structures. You have organelles. You have the nuclear envelope, the endoplasmic reticulum, also known as the Golgi apparatus, lysosomes, vesicles, vacuoles, and of course plasma membrane. So in general, this system serves, but is not limited to, right, synthesizing proteins, transporting those proteins to membranes or organelles out of the cell or within the cell itself. Okay. It also serves for metabolic activities and the movement of lipids and detoxification of poisons. Depending on the cell type, the membrane and the endomembrane system and the membranes involved could vary in the composition, the proteins that are embedded within a particular structure, and of course the function that it needs to do. So we move on to the endoplasmic reticulum, ER, which begins this whole biosynthetic factory processing, shipping system. So if we look at this diagram here, this is how the endoplasmic reticulum looks like. Okay, so it's a membranous system of interconnected tubules and flattened sacs. These flattened sacs are called cisterna. Okay, so imagine you get a balloon, for example, and then you squish that balloon into a little flat pancake. You see these structures here. Okay, so all of these flattened sacs are referred to as cisterna. But you can also have tubular structures. Again, the same membrane, but in a different form. All right. So again, interconnected tubules and flattened sacs. ER is also continuous, so it's coupled to the nuclear envelope. All right. So you have the nuclear envelope in purple, and then you have the ER in blue, in this dark bluish color. Within the ER, the fluid is referred to as the lumen or the cisternal space. Okay, the rough ER, it is studded with the outer surface of the ER is pretty much covered with ribosomes or studded with ribosomes, and that's one of the main distinguishing features between the rough ER and the smooth ER that we're going to take a look at. So usually the ER is connected to the nuclear envelope, as you see in the diagram. And if we were to see an electron micrograph, it would look something like this. You can see the flattened sacs in that little space, the cisterna. All right. You can see these little dots. These little dots are the ribosomes. They're either embedded within the membrane of the ER or floating around. And here, these are little cross sections of the tubular section of the smooth ER. Okay. So let's take a look at some of these characteristics of the ER. As I mentioned before, it's an extensive network of membrane. It makes up over half of the total membrane in the eukaryotic cell, or most eukaryotic cells. The term endoplasmic, meaning within the cytoplasm, and reticulum, referring to a little micro net or a little net. Okay, it's again, this is something you need to memorize. That space that is inside the sac is referred to as the lumen, the ER lumen. It creates that compartment separating itself from the cytoplasm, or the cytosol more specifically. Again, there's two types of ER, the ER and the rough ER. The smooth ER, pretty much the name tells you it has a smooth surface because it lacks ribosomes on the surface, and the rough ER is studded with ribosomes, giving it a rough appearance. And it's the rough ER that is continuous to the nuclear envelope, to the outer membrane of the nuclear envelope, which also has ribosomes. Let's take a look now at some functions of both of these Okay, so I put them side to side. This is a good question for comparing, contrasting on any test or a quiz, right? The functions between these two types of ER. So talking about the rough ER in terms of their function primarily, right? So one of the main things it does, if not the main thing it does, is protein synthesis and secretion. Okay, so the ribosomes that are embedded on ER, they produce proteins to be secreted throughout this endomembrane system. Some examples of what can be produced is, for example, the pancreatic cells, they synthesize and secrete insulin, but no matter what protein you're producing, the polypeptide chains that are being threaded and created from the ribosomes, it's happening in the ER, and moreover, it's being threaded into the ER lumen, right, or the compartment space through pores, membrane pores. And remember when we talked about proteins that they have different structure and levels of structure, the functional shape, so the tertiary or even quaternary structure, occurs and folds within the ER lumen. Okay. Most of the secretory proteins that are being made are glycoproteins, and basically meaning that there's carbohydrates that are being attached to that protein by the membrane enzymes, ER membrane enzymes. Okay, so it works for protein synthesis, protein transport. The ER membrane keeps the secretory proteins separate from cytosolic proteins, or proteins that are needed within the cell itself or within the cytosol. So these secretory proteins, they're going to exit the ER in packages that we call them vesicles, right? And these little ships, they bud out from the ER membrane onto the next organelle that we're going to talk about. Okay. More specifically, these little vesicles are referred to as transport vesicles because they carry proteins from one area of the cell to the other, to another area of the cell. So aside from protein synthesis and secretion, you have protein transport, and you also have membrane production. So rough ER functions as a membrane factory. Remember, when I talk about membrane, I'm talking about the phospholipid bilayer membrane. So it acts as a factory, growing by adding membrane proteins and phospholipids. So it's connecting all these pieces together to make more membrane and enlarging, getting bigger, creating these vesicles. These vesicles ship out. They can be added to other membrane areas. Okay, so membrane proteins, they're inserted into the ER membrane, and they're anchored by hydrophobic regions. Remember what the hydrophobic regions were, right? Areas that don't, associate well with water, so they stay in internal in terms of proteins, membrane proteins. Okay. Rough ER also produces phospholipids, assembled by enzymes in the ER membrane. Okay, to grow itself. That's important to note. To grow itself and enlarge. Okay, so the ER membrane expands with proportions transferred via transport vesicles. Again, vesicles throughout the endomembrane system. Functions of rough ER. Smooth ER, okay, functions primarily in lipid synthesis. Enzymes within the smooth ER, they synthesize different types of lipids, oils, steroids, and phospholipids also. Okay, they produce steroid hormones like sex hormones, adrenal gland hormones. Okay, so you're going to have a lot of smooth ER in structures that would produce sex hormones. For example, like the testes and the ovaries would have more abundant smooth ER. Okay, so that's something that you need to think about when you're talking about organelles. If you have a structure that is doing more of a particular thing, you're most likely going to have more of that particular structure. In this case, more smooth ER to produce more hormones, more steroids. Okay, the same thing goes for mitochondria that we're going to talk about a little bit later. Okay, so lipid synthesis, Detoxification, smooth ER enzymes detoxify drugs and poisons, especially in liver cells. So you're going to have more smooth ER in these liver cells. Detoxification, how does it work? It basically adds hydroxyl groups to make drugs more water soluble so they can be excreted in the out of our urinary system. Okay, there's some drugs, barbiturates, if I'm saying it correctly, barbiturates, which are drugs that are involved with relaxation and sedation, right? Alcohol and other drugs induce smooth ER proliferation. So the more smooth ER you have proliferating, you're going to increase detoxification capacity, all right? Now that is a problem because increased detoxification leads to drug tolerance. So you don't want to get too used to a drug because you're going to immediately process it quicker, right? Requiring you to have a higher dosage of the drug. And there are some drugs that could have potential side effects. So, overdosing is not a good thing. Okay. Broad-acting detox enzymes, they can reduce the effectiveness of other drugs. Again, barbiturate abuse or this type of sedative, taking too much of it can affect the use of antibiotics just because of how they have the potential to interact with each other. Okay, so detoxification, calcium ion storage. So you have, okay, smooth ER can pump calcium ions from the cytosol into the lumen. Remember, we have the cisterna. The lumen is the space inside the cisterna. Okay, why is calcium ions important? We're going to see this in the later chapters. In muscle cells, nerve impulses trigger calcium release, which leads to muscle contraction. So you're going to need a lot of calcium to be creating phenomenon, this overall movement of muscle. In other cells, calcium release can trigger responses like vesicle secretion. So it's always good to have a certain concentration of calcium ions based on what you need to do. Okay, so rough ER, smooth ER functions. Let's talk about the next organelle, which is the Golgi apparatus. So take a look here. If we kind of skim through the diagram here, it's right beside the ER. Looks quite similar in terms of the flattened sacs. Again, you have cisterna. You have these flattened sacs with the compartment spaces inside. What's happening in this case, vesicles that are formed from the ER, they're going to be shipped and moved toward the Golgi apparatus. If we take a look at the micrograph, you see all of these little lines sandwiched together, right? Where you have one side and then you have another side, you have a receiving side and a releasing side that we're going to take a look at what is happening. Okay, they have a specific name. It's called the cis side or the cis face, and then you have a trans side or a trans face. Golgi apparatus. So this is the shipping and receiving center. Depending on what textbooks you use, this is also called the Golgi complex. Either or is equally fine. Okay, so main function is to process and distribute the proteins and other molecules that are synthesized from the ER. They're labeled to go to their final destination point, if I can say. So they modify, stores, ships cellular products, especially in secretion specialized cells. Taking a look into the structure. Be familiar with this idea of the flattened sacs. They're stacked flattened sacs. Each cisterna has its own internal environment separate from the cytosol. Again, similar to the ER. Vesicle transport materials between Golgi compartments. So the vesicles transport the materials between the Golgi and other places around the cell. In terms of direction, Golgi has, again, a receiving side and a shipping side. So the one that is closer to the ER is referred to the cis face. And that's where the vesicles are coming in and they generally fuse with the Golgi membrane and they liberate whatever contents they have in there into the compartment spaces. As the material is moving, right, the little sacs are also moving, right, and they're going like in an assembly convoy. And the cisterna themselves are maturing. as the protein is being fixed or whatever molecule is being processed. So ER vesicles fuse to the cis phase. They transfer the content into the Golgi compartment. And then after that whole preparation and manipulation of the protein is finished, the trans phase will generate a vesicle that would carry the modified or that final product elsewhere. What could be some modifications? You have proteins, such as glycoproteins, though they're going to be undergoing further carbohydrate modifications. More carbohydrates could be added to the protein. Some sugar monomers can be removed or replaced with another monomer, sugar monosaccharide. So you can have different types of carbohydrates embedded within a protein. You can also have membrane phospholipids that are being chemically altered and tagged, right, depending on what particular thing they need to do. Modifications. And then you have your manufacturing role, where you have the Golgi producing macromolecules, again, like polysaccharides, directly just stemming. You don't necessarily need to have a protein, but you can have a polysaccharide just stemming out of the cell surface, okay, of the cell membrane surface. You can have in plant cells, the Golgi apparatus synthesizes pectins, okay, and other cellulose polysaccharides for, let's say, reinforcement of cell wall. Transport and targeting, so a little bit more information on this. So Golgi products, they're sorted and tagged with molecular markers. They could be phosphate groups to help in the tagging for correct destination, depending on where they need to go. Okay, transport vesicles, they bud out. So when you have that product, the vesicle will, going back to that little picture here, you can see how there's a formation of the little vesicles stemming out from the edges of the cisterna, ultimately until they nip out, right? They bud out. They're removed into this little sphere, this little vesicle that will move somewhere in the cell or outside of the cell. Okay. So again, transport vesicles, they bud from the trans phase, carrying cargo to organelles or the plasma membrane if they need to be embedded, if they're, let's say, a peripheral protein or a transmembrane protein and whatnot. So external molecules on the vesicles help them dock at particular target locations. Okay, we're going to see this definitely a lot later in the later chapters. Going a little bit back about that cisterna maturation. So there's a dynamic model of the Golgi, which is initially thought to be a static structure, right, with vesicle transport between cisterna. But recent research suggests that the sacs themselves undergo maturation, and they're moving and modifying as you're going throughout the Golgi complex. And as they're moving themselves, they're also modifying the cargo as they progress from the cis side to the trans sides. So probably both things are happening. They probably move some vesicles from one to another to another to another, or maybe they're going together, the cisterna plus the vesicles plus the cargo, the products, the material that they're altering. Okay. All right, let's continue on to certain types of vesicles. Again, within this transport endomembrane system, you have lysosomes, very specialized sacs, vesicles, right? So these lysosomes are the digestive compartments of the cell. Now, what does that mean? They're filled with hydrolytic enzymes used for digestion, breaking. Okay, so these enzymes, they function best in acidic environments inside the lysosome. So they're very special. They have a very, very unique environment compared to the whole cell itself. Okay, very acidic. That's an important thing to note. If lysosomes ruptures, cells become inactive in the near neutral cytosol, but excessive leakage can cause self-digestion of the cell. So basically the cell can break apart. Formation and structure, hydrolytic enzymes and also lysosomal membranes are produced by the rough ER. Okay, so important to note with that. They're produced in the rough ER and processed in the Golgi. Some lysosomes form by budding in the trans phase of the Golgi, okay, and they're protected from self-digestion by their 3D structure. All right. So here's a little depiction of these next two forms of intercellular digestion. You have one that is called phagocytosis. And phagocytosis occurs, many organisms and cell types can do this, where they engulf, so phago meaning eating, they engulf food particles into vacuoles, and these vacuoles, they would fuse with lysosomes to break the food particles down. When you break the food particles down, you get basically the building blocks, right? You get sugars and amino acids that could be absorbed later by the cytosol and be used to construct different types of macromolecules. Macrophages, very, very important cell for the immune system. It's a type of white blood cell, and it is used to destroy bacteria and foreign invaders, right? So it's a very important cell type, phagocytosis, right? So this is a little depiction of phagocytosis. You have your little, food particle that you want to bring in. This creates a vesicle as the particle is coming in. It's encased by a food vacuole. That food vacuole fuses with the lysosomes. The hydrolytic enzymes begin to digest, molecularly digest, right, the food particle, and then you get the remaining kind of simple molecules there. Different cell types have a certain amount of lysosomes. In this case, the macrophages that I was talking about, look how many lysosomes are there, right? Because their main function is to destroy particularly, bacteria, pollen, and so forth. Sorry about that. Then you have autophagy. Is a controlled way, right? Components. Okay. Next is autophagy, where lysosomes recycle damaged organelles and cytoplasmic material. What happens here? A double membrane surrounds damaged components, right? And the lysosome fuses to break down the contents. Okay. So this is important because the cell can reuse digested materials to, as building material and renew itself. So you don't have to waste a lot of material and be creating new and new material again and again, right? If you have this kind of recycling center. So the human liver, right, and the cells within the liver can recycle half of their macromolecules each week. So that's quite interesting how efficient the cells can be in recycling their own material. Here's an example of autophagy, right? You have a lysosome that is fusing with a vesicle that is containing damaged organelles. Okay, so you have your, let's say, a mitochondria that's been worn out, that's been physically damaged. You have this vesicle that is coming in with a lysosome, and that lysosome is coming in to digest the material. There's an additional little vesicle that we see here, a peroxisome, that we're going to talk about in a little bit, dealing with H2O2, with hydrogen peroxide. Here's a micrograph, again, of the same situation there. You have a vesicle containing two damaged organelles. So the interesting thing about the vesicles is that they can change. They don't have to necessarily be just tiny protein or molecule level transportation. You can have organelles within themselves, right? Some diseases relating to lysosomal storage, very important, right? There's some diseases caused by a defective lysosomal enzymes, missing or defective, which could lead to the accumulation of undigestible substances, right? So imagine you have waste that you need to remove at the cellular level, but you can't produce the enzymes to break them apart. An example is Tay-Sachs disease, right? So it results from a missing lipid digesting enzyme, which causes lipid buildup in brain cells. These diseases are rare, but can severely disrupt cellular function and cellular metabolism, right? And lastly, next up is vacuoles. So they have a variety of functions. There's different types of vacuoles that we're going to take a look, but diverse maintenance compartments. All right. So vacuoles in general, they're generally large vesicles. They're derived from ER and Golgi apparatus, developed. They are part of the endomembrane system, and they have very, very selective membranes that regulate solutes. So that's a key important thing to note, right? Solute transport. I don't want to say harvesting, and maintenance, storage. So here's some examples. Food vacuoles. They're formed by phagocytosis. They aid in intracellular digestion, right? So they bring in food particles for a cell to come in and eat, especially single-celled organisms that need to break down some form of food. Okay. Contractile vacuoles, right? They're found in freshwater protists, okay, like paramecium. They can pump excess water out of their bodies to maintain ion balance. Okay. We're going to see that a little bit later when we get into osmosis. Hydrolytic vacuoles, they're present in plants and fungi. They perform enzymatic hydrolysis. So they're similar to animal cells, the lysosomes of animal cells. All right. You have storage vacuoles. Again, like I mentioned before, they can serve to contain many different, you can see these in plants especially. They could store proteins and seeds, right? So they have high capacity to store. Defensive vacuoles, they contain toxic or unpalatable compounds to deter herbivores, again, with plant cells, all right? And just to kind of finish here, pigment vacuoles, so they can store pigment like red and blue in petals to attract pollinators, again, for plants. Then you have the central vacuole, an incredibly large vacuole within plant cells, right? They're developed via the fusion of smaller vacuoles. So if you take a look at the diagram here, incredibly large, even surpassing the nucleus, right, in terms of size. So they can store inorganic ions like potassium chloride ions, salts. And it's very important for plant growth, regulation of water, okay, and salt balance. So whenever it needs, it can modify the size of the cell. It can expand with minimum cytoplasma increase. So these central vacuoles, they help maintain the cell structure and sufficient plasma membrane, maintain an adequate surface area relative to the cytosolic volume. So if you take a look here, the cell's generally large, but you still have, sufficient cytosolic volume to maintain the necessary metabolic activities, but you can increase in size. You can maintain the cell shape in the case of plant cells becoming turgid, right? And that's one of the key features that enable plants to stay upright and nice and vibrant. Okay, so taking all these together, we get this review, a summary of the endomembrane system, where you can see how the movement of a protein that has been synthesized goes throughout several organelles until it's basically excreted or where it needs to go. Of course, every step requires some modifications, so the protein can be used, or that product can be used effectively. Okay, so again, we start from the nucleus. The nucleus, we're going to see later on the instructions to produce a protein via transcription and translation. That protein being synthesized in the ribosomes and the roughly as they're being created, vesicles will ship that protein to the Golgi from the cis side, having further modifications until it moves out through the trans side on another vesicle to be shipped out, right? Very good. So that finishes the first video of chapter 6 on cells, structure, and functions. I will continue with section 6.5 on mitochondria and chloroplast. This will be part 2 of chapter 6. So thank you for watching. I hope this video was useful, insightful, and I'll see you in the next one. Bye-bye.